So this is a secondary analysis from the QuANTUM-First study, and a key secondary endpoint of the QuANTUM-First study was to determine the percentage of patients who had achieved CR, how many of those had cleared MRD, how many of those were MRD negative. The entire field of AML is shifting its focus onto MRD and different techniques. And that is a question of interest just to start with. Can we use MRD in a prospective trial to predict outcome? But in addition, this is the first use of, I think, a novel approach to detecting MRD. It is not flow cytometry based, which is very challenging in AML, despite all the controversies of who's in favor or not. This is a very specific test, and I want to stress it's not a new test. It was first developed by, I think, Michael Heuser's group 10 years ago, and used by the Alpha group 3 years later, and used by 2 other groups a few years later to look at very specific FLT3-ITD-specific MRD. PCR-NGS, it's a double assay, and you can actually order the assay from a diagnostic lab right now, and a third-party payer will pay for that. And there are centers, including my own, that are currently using this. This was the first test. This abstract presented the data on the first test of this technology, or this approach to MRD, in a prospective clinical trial. We've just demonstrated that it is associated with better survival, and that the specific drug treatment lowers that specific MRD, so it's a specific effect of the drug. So it's an important abstract, but just a first step.